This is Mia and Drayton's presentation on Anna and her E. coli infection.
Theodor Estrich was a German-Austrian pediatrician who discovered E. coli in 1885 while studying infant feces for the causative organism of fatal intestinal diseases in children. He isolated a quick-growing, rod-shaped bacterium from infant fecal samples and named it Bacillus communis coli. But after his death, it was shortened to E. coli. He used his own anaerobic culture methods and another scientist's staining technique to isolate a variety of bacteria from infant samples. The prefix Escherichia comes from the last name of the pediatrician, who is Theodor Estrich. The suffix coli is derived from the place in the body the microbe is commonly found, in the colon.
For the process of observing this microbe, first we're going to fix the specimen from the slide to the stage, and then we're going to locate it with a 4x objective lens. Once we've gotten a good view, we can then locate further with switch to the 10x objective lens and start identifying colonies that we wish to look at. And then we switch to the 40x objective lens to further isolate a colony to look at. And after that, we apply immersion oil to the slide, and the purpose for this oil is to increase the resolving power of a microscope by immersing both the objective lens and the specimen in a transparent oil of highly refractive index. After that is done, we'll switch to the 100x objective lens for a total of 1,000 magnification, and then we can observe the color of the microbe, which should appear pink, the shape of the microbe, which is rod-like, and the arrangement of the microbe, which is single cells.
The expected results of the Gram stain that we are going to expect to see is that the Gram stain's negative because of the thin peptidoglycan cell wall. Colonies cultured from sorbitol appear pink due to the fermentation process of the microbe in the culture. Non-pathogenic strains of E. coli will be able to ferment the lactose in the sorbitol, producing acid. Pathogenic strains of E. coli will not be able to ferment in the sorbitol culture. E. coli stains gram-negative as well because it has an additional outer membrane that is composed of phospholipids and lipopolysaccharides. The presence of lipopolysaccharides on the outer membrane of bacteria give it an overall negative charge to the cell wall. In the case of gram-negative bacteria, the alcohol dissolves the outer lipopolysaccharide membrane of the cell wall and damages the cytoplasmic membrane to which the peptidoglycan does attach.
For this specific strain of E. coli, STC0157, the symptoms come from the production of toxins from the microbe that weaken the lining of the intestines. This causes inflammation and leads to bloody and slimy stools as they pass, and many fluids are lost in this process. And many patients often experience stomach cramps, to which Anna also had. In some cases, vomiting can occur, and as well as a fever. It is recommended to drink plenty of fluids and electrolytes to resupply what the body is constantly losing through passing and trying to get rid of this infection.
In order to resolve Anna's infection of STECO157, it can resolve on its own if treatment care is thoroughly followed through, but at this time no drug treatment options can be prescribed. Proper care includes plenty of fluids and electrolytes due to the loss from diarrhea. Since Anna is so young and antibiotics are more harmful than beneficial, we're not going to prescribe any antibiotics as they can lead to further complications. This can lead to the development of C. difficile, which is the infection of a large intestine or the colon caused by the bacteria Clostridium difficile. This is due to antibiotics that fight bacterial infections by killing off the so-called bad germs. They can also get rid of the good germs that can fight off harmful infections, thus leading to C. difficile. Possible disruption in development of the kidneys and livers can also arise. We're going to closely monitor her throughout her recovery because she is so young. I'm sure her mother will keep a very close eye on her. If symptoms also worsen after expected recovery time, intravenous or otherwise known as IV drips may be recommended to ensure dehydration isn't a factor.